Okay, welcome to Let's Get to the Marks. I'm going through a couple of biology maths questions for A-level biology. Hello, obviously. Right, let's get on with it. What I suggest is you pause the video, have a go at the question, then unpause and watch me go through the explanation. Of course, you'll need a pen, paper, and a scientific calculator for these ones. All right, let's get into it. An air bubble was introduced into the glass tubing in figure one. Here's figure one. The air bubble moved a distance d of 1.5 centimeter cubed in 120 minutes. The radius of the lumen hole of the glass tubing was zero point 0.6. So the radius is zero point 0.6. It's moved 1.5. Use this information and the formula pi r squared times d to calculate the rate of water movement in the glass tubing in centimeter cubed per hour. Use 3.14 in your calculation. Okay, let's get into this one. What we want to be thinking about here is they've told us D, they've given us R there, and they've given us pi. So that is all beautiful. But they've also given us centimeter cube per hour and 120 minutes. All right, let's get on and carry out the calculation. So we're going to do 3.14. You're going to times that by the radius, which is zero point 0.6. We want to square that and then times that by the distance, which it told us already was D. Beautiful. All lovely jubbly. So what's that? 3 point, that's basically 0.36. Is that 0.36 or 0.03? It's 0.36. And if I times that all together, we're going to get like 1.6956, something like that, centimeter cubed. Now, you could go and slap that down there, but you won't get the mark. It's a peasily little 1 mark, and you won't get it for that. So you have to take into account that they've asked for rate. So always check the units. They give you a clue as to what you're going to do. So I always like to say, check units. They give clues as to the calculation or to part of it. They normally give you some sort of clues. So we need to divide centimeter cubed by hours. When you get this to the -1, that means that is the same as writing centimeter cubed back in GCSE days, like that, centimeter cubed per hour. That's what it means when they write to the -1. So we need to divide this distance by the hours, and we've got 120 minutes. So 120 divided by 2, you don't really need to do that, but that equals 2 hours. Well, no, it doesn't. That would equal 60. So 120 divided by 60 minutes in an hour equals 2 hours. So basically, we need to divide that by 2, and our answer is going to be 0.8478. You could probably round that to whatever you like. It doesn't give us 2 sig fig or 2 DP. So you could round it to 3 significant figures at A level or 2 decimal points places. Lovely jubbly. That's going to get you just 1 mark. They are so stingy. Right, let's move on. Okay, 2.1. So we're going to have a go at this one now. 2.1 shows an image of a red blood cell at a magnification of 5,500. Look, I put a funky little red blood cell, erythrocytes up there. Anywho, calculate the actual diameter in micrometers of the red blood cell between points P and Q. Show your work in 2 marks. Why don't you grab your calculator, grab a pen, grab some paper, pause the video, have a go. Okay, what we're going to do here is you can see they've asked for the actual diameter and we've got magnification. This would ring little bells in your head and you should think of the old I am equation. And we want to work out A, so we want to do I over M, and that is going to give us A, which is our answer. Now, we've got the magnification, so we want to do I over 5,500. Now we just need to work out I. I, obviously, image. Guess what this picture is? It's an image. A red blood cell's not that big. So anything you're measuring with your ruler, a little clue if you're ever getting confused. Am I measuring the actual or the image? If you're using a ruler and they're asking you to measure something, you must be measuring the image. We're talking about microscopy here, microscopic images. So you couldn't be measuring them with your ruler without some sort of graticule and things. So yeah, this is going to be the image. And so when you measure that, you get about 40 millimeters. Now, do beware if you're doing a testing class and your teacher has done a magnification question where you have to measure something, often they will shrink down the image for the exam paper. And often you'll be there with your teacher baffled as how to get to the correct answer. And the answer is that they have not used the correct size that you got in the exam. So In the exam paper, this equaled 40, 41, 42 millimeters. So I'm going to give it 40 millimeters here. So what do we do next? Millimeters on our ruler. You could have measured this in centimeters, but then you've got to go from centimeters to micrometers. I prefer to stick with millimeters because it's a straight um, going in divisions of 1,000 meters, millimeters, micrometers, you're going in thousands. So 40 millimeters, we want to change that into micrometers. And so we're going to 40 times 1,000. We have 40,000 micrometers. That's our image. And we're going to divide that by 5,500. And that's going to give us something along the lines of, what is that? 7,777.27 is what I get on my calculator. I don't know what you get, but yeah, there'll be an acceptable range in the mark scheme because not everyone will measure that to 40 millimeters. Some people will measure it to the end of these arrows. Some people will measure that little gap there, the little bit there. So there's going to be a range of a couple of millimeters they're going to allow. So lovely jubbly. That'll get you 2 marks. So 1 mark for the conversion and 1 mark for putting the numbers in and getting the answer. Wunderbar. Let's move on. Right, we've got another one here. A Hey, a hemocytometer is a special microscope slide that can be used to determine the mean number of red blood cells in 0.004 millimeter cubed of blood. A researcher prepared a 10 to the minus 3 dilution. So let's start circling things. We got that. We got a dilution, 10 to the minus 3, of a sample of blood from an adult using a hemocytometer. The research determined the mean number of red blood cells in zero point 0.0. So the mean number was 21. So we got 21 red blood cells in 0.004. Okay, that's how many blood cells are in 0.004. The volume of an adult was 4.8 decimeters of blood. Calculate the total number of red blood cells in the body of this adult. 2 marks. Pause the video. Have a go. Okay, I'm going to go through the answer now. So what we want to do first of all is recognize that we've got 2 different volume units here. We've got millimeter cubed and we've got decimeter. So I'm going to change this decimeter into millimeter cubed. Now, if you're a chemist, you're used to changing decimeters into centimeter cubed, and that is times 1,000. But to change centimeter cubed into milli, we're going to times another 1,000 because a centimeter is 10 millimeters. Cubed means 10 times 10 times 10. So it's 1,000. So we're going to do, we're timesing a million basically. So you could just learn that to change decimeters to millimeters, you times a million. That is also optional. So we're going to have 48 million millimeter cubed in our adult's body. We want to know the total amount of red blood cells in that adult's body. Well, we know in a diluted sample, diluted blood sample, there's 21. So let's work that out. That means 0.004 contains 21. So I want to find out how many 0.004s go in that. So I'm going to divide by 0.004 and then I'm going to times, there's 21 in each of those 0.004s. I want to find out how many 0.004s are in 48 million. So I'm going to do 48 million divided by 0.004. That gives me 12 billion. There are 12 billion samples of 0.004 in 48 million. So we've got 12 billion of those. However, I need to times that by 21. That's going to give me 252 billion. And this would get you 1 mark. 1 mark, you say. Why isn't that all the marks? That is 52 billion red blood cells in the body of an adult. But that's not true. We were using the diluted sample. Now, they told us that they diluted it, and this 0.004 was diluted. So actually, we would need to times by 1,000. It was diluted 1,000 times 10 to the 3 or 10 to the minus 3 was diluted. So I need to times this by 1,000 to get it into a non-diluted sample. And that is going to give me 252. There's too many zeros. And that is going to be 12 fat zeros. And if we put that in standard form, it's easier to write 2.52 times 10 to the power of 13. And that is your answer. So you may have forgot to do that. That would get you the mark. If you did any of this working out here, you're going to get at least 1 mark. All right, let's move on. Okay, final question in this little maths pack. By the way, you can check out the playlist and you will find I've got loads of biology maths calculations done. You should check them out and work through them. Lots of people struggle. The only way to do is to do a concerted amount of maths all in one go. So make sure you put aside half an hour every couple of weeks for just doing maths questions and you will become an expert. It's not something you can just do the odd one when you're doing a past paper and you're going to become an expert at them. You need to put aside a whole chunk of time because then you'll see patterns and similarities and conversions and techniques you need to use. Okay, the student used a sterile pipette with 0.1 centimeter cube graduations to transfer 2 centimeter cubed of B subtilis, that's some sort of bacteria, into a test tube. What is the uncertainty in measuring 2 centimeter cube with this pipette? So calculate the percentage uncertainty of this 2 centimeter cube measurement. 2 marks. So first of all, the uncertainty. Well, you need to know this equation off by heart. There is an uncertainty equation, and the uncertainty equation is smallest division. So the smallest kind of measurement or on your scale, which in this case is 0.1, and you divide it by 2, and that will give you the uncertainty. So dead easy. We go 0.1 divided by 2, and that gives us 0.05. And that's because essentially when you're measuring, you never know when you're measuring something whether it's on that line or that line. So it can also always be either there or there. It can be 0.05 out, basically. It can be a little bit out. So, and that is everything will always have an uncertainty of half the smallest division. Anywho, that's 1 mark. Right, we need to do the percentage uncertainty. There's a calculation for that. And the percentage uncertainty is just your uncertainty divided by the measurement, right? And then times 100. So we're going to do the uncertainty over the measurement times 100. And the uncertainty is 0.05. Anytime you use this pipette, there will be an uncertainty of 0.05. The measurement we made is 2. So that's going to give us like, that divided by that is going to give us like something like 0.025. And then we're going to need to times 100, and that's going to give us 2.5%. Now, just another question that you may be asked at some point, how can you reduce the percentage uncertainty? Evidently, if we increase this number at the bottom, we're going to reduce the percentage uncertainty. So instead of us transferring 2 centimeters to each tube, why not transfer 4 centimeters to each tube? And then you're going to reduce the percentage uncertainty. It's going to go down to 1.25%. So when using this pipette, you could reduce the percentage uncertainty by transferring a larger volume. So generally in science, the larger amounts or masses or volumes that we're measuring, we're going to reduce percentage uncertainty in our results. All right, guys. Make sure you check out the other maths videos I've got for A-level biology. Don't forget sharing, caring, hit the like, punch up the bell, subscribe. Let's get to the marks. I'll catch you guys on my next video.
You're still here? It's over.